Okay, cool. Sorry that took 1 minute longer. Um, so let's continue on and proceed with the next part of the pipeline. I'm going to show you again the Seurat pipeline so that we know where we are right now. Might seem a little bit like we are, behind time, but don't worry because we just finished quality control, but the next few parts are actually pretty fast. I just because we sort of spend a lot of time on understanding the object, we understand how to create new columns and look at the quality control. Now this is going to be a little bit more faster. Cool. So we're gonna do a linear reduction of the dimensions. I'm going to explain what is that. I'm going to do clustering and then perform these 3 downstream tasks towards the end of the class. All right. So if you have completed running SC transform, it should look something like this. I'm showing my RStudio now. So it should have completed with this, at least this line that says set default assay to SCT. And then it says that there were 50 or more warnings. Don't worry about this. This is internally, I've checked the warnings beforehand. This is nothing detrimental to the downstream, unless this is some issues with the namings and some internal stuff. So don't worry about that. But then this message here tells you that the default assay has changed. And this is what I'm trying to say earlier on, whereby if you were to print out the, let me, keyboard is again skipping. Okay. Okay. So if you type default assay followed by seurat.merge and run this, you can actually see that instead of RNA, which we have actually print out previously, it prints out SCT. And the reason is because this SC transform will create yet another assay within your Seurat object. And now it shifts or it ports all the data from this Seurat the initial Seurat pipeline into SC transform assay, this new SCT transfer, SCT assay. But don't worry because it's all an internal object. The porting is seamless. Everything will go on as expected. So you can continue on with the pipeline as though it is a RNA assay as previous. Okay. So we will now perform next part, which is dimensional reduction. I like this part a lot, mainly because it looks very pretty. You plot out very pretty scatter plot. We're going to start with the linear dimensional reduction. And the difference is that there's different model that you can actually use to reduce the dimension. What does it mean by reduced dimension? So if you imagine your cells as a dot in space, right? So if you have like a graph and you point out or you kind of plot out your cell, it's difficult to pinpoint the exact coordinate because you have, in our case, 28,000 genes. And how are you able, or how do you assign the weights for each of these genes to identify the distance or the correlation between two cells? So, and then plotting it out based on this number of dimensions is actually impossible. So the idea behind dimensional reduction is to compress this vast information that you have, and so that it will now be reference based on at least a two-dimensional axis. So it can plot it on an x-axis and a y-axis, and you can actually also compare these coordinates between two cells to see which cells are closely related to each other. All right. So summary, very big matrix compressed into just x and y values. There are several ways to do this. The most straightforward way, I guess, theoretically easier to explain is PCA plot, and it's a very common plot in a lot of publication. You also have t-SNE plot, is stochastic way of modeling this reduction, and you have UMAP, which is currently one of the standards for single-cell RNA-seq, because it gives you better result. I'm not going to give you a lot of details about this. I'm going to give you a link if you want to have a read on the details, but basically PCA is what we call as a linear reduction, because it looks at the linear relationship between the principal components between two genes, and then plot it out based on that particular component plus the orthogonal component. All right. t-SNE and UMAP looks at nonlinear reduction, and it basically looks at the probability of the distance between two cells, and it sort of like retains this distance from the highest dimensional all the way to the lowest dimension. So it's slightly a bit more complex, but you can see the difference when I plot out the PCA and the UMAP in a while. All right. So we're going to do PCA plot first. Usually PCA, well, you can choose which one to plot. Like you can start off with you can choose that you want to do UMAP or t-SNE, but a lot of these functions actually rely on the initial data from PCA, because PCA gives you this compressed data frame, these compressed principal components that will then be built upon to generate the t-SNE and UMAP. So we will run the PCA reduction first and visualize it, and then followed by the other two dimensional reduction. Okay, so PCA, let's just start PCA first. We will again overwrite our previous Seurat object, so Seurat.merged. And the function to perform PCA reduction is run PCA. Again, code will be in Etherpad. And the arguments will be the object Seurat.merge, and we will have to specify the features, which genes do you want to use as the initial list of genes to consider for principal component analysis. So features here, very conveniently, the developer of Seurat allow you to provide functions for this variable. So we're going to type it as it is, variable features object equals to Seurat.merge, like that. All right, again, code is already in Etherpad, so you can copy and paste this and run the code in console. Will take a few seconds, but not too, it's not going to be too long. I'm not sure whether this is running on multi-thread. So for me, it will be relatively fast. But if you're on a laptop with 4 cores, it will not be like exponentially long, but it will take slightly longer if they use multi-threading. So mine is done. Give me a thumbs up if you have successfully run PCA analysis on your computer as well. Cool. Cool. I think we are around there. I can see whether Katie, did you give a thumbs up? Are you okay with the object? Yep. Okay. Cool. Okay. Cool. Then we can visualize this in on a scatter plot. The thing about Seurat is that previously we plot this feature scatter, which plots out the graph that you see in our studio right now, the feature counts and the gene counts and stuff. They do have specific functions for other plots. So in order to plot out the PCA or UMAP, it uses this function called dimplot for a dimensional plot, and it plots out the two principal components on a scatter plot. Essentially, that's it. Right? There will be a couple of other functions which also plot out this dimensional plot, but then it adds on the additional information like expression and stuff. So I'll show you that function in a bit. But let's run dimplot followed by Seurat merge and see what do we get over here. Okay, run this in console, and you get this. right? So each cells are labeled as cerebellum because remember we have preset or we have assigned cerebellum or the region column as the default identity of all our cells, so it will be labeled as such. We can also see dots on this plot. Each dot represents one cell, and the coordinate of this of each cell is relative in space to the other cells. So majority of the cells are at this bottom half of the plot. You do see some clusters, orthogonal clusters that's going downslope here. You can try to figure out what these cells are if you want to sort of find the features of these cells. But we're not going to do that. One thing that you realize from this PCA reduction is that cells are still very condensed. They are not separated well enough. Like it's difficult for you to figure out how many cells are there in this cluster, what are the identities, because they're basically so close that they are almost overlapping to each other. They're not overlapping. Each cell has its own coordinate, but they are so close that it's difficult to tease apart the cells, right? I mentioned that that the cells are labeled by the region, cerebellum. We can basically plot out or group them using our different groupings from our metadata very quickly using dimplot. So instead of renaming the identity, we can provide this argument called group dot by, and specify the name of the column that you want to use to group your cells. So in this case, let's use auric.ident. This is the very first column that was created, and this should have the values E14, E16, E18. And let's see what, yes.
It's a short question. So in my
plot, yep.
On the y-axis, I have 0 to minus 150. So you have 150 to 0. And on my y-axis, it's 0 to minus 150.
Yeah. Everybody, I know there's a reason for it. But who else has this inverted scale?
I have the same.
I see. It should be the reason is one of the reasons because PCA is it will put it this way, put it simpler way. It samples your data set and create this plot every single time. So whenever you do PCA a couple of times, usually some things might be slightly different. The other thing could be that the scaling or the values of the gene expressions is slightly different, but I don't think that should differ because we are using the same cow matrix, we did the same pipeline, and it shouldn't differ. Is the shape of your PCA plot similar?
Yeah, it's basically inverted.
Yeah, mine too. So it's just
kind of mirrored on both axes in a way.
The other reason could be the version of the Seurat. Let me see my version. I remember I installed the development version. So if I go to packages and look at Seurat, mine is 4.0.2. If somebody can quickly look at your version and tell me, is it 4.0.2?
I have 4.0.3.
0.3. Probably that could be the reason. It's the end. Just now, Rewang, I think you have the same plot. Do you have 4.0.3 as well?
Yes, I have 3.
Okay, so then it could be that the code that you have in 4.0.3 is slightly updated, and mine is they changed the code so that it represents a different scale, but essentially the coordinates are the same.
Yeah. Okay.
Right. Yep. So it's not something dire, just a change of code, or so you see different scale but same coordinates. Okay.
Okay, thank you.
Good you pointed out, because I wouldn't pick that out if I were to run this on my own. And it's correct. I mean, the plot is correct. Just that you didn't think that the scale, they would change the scale like that. Cool. Thanks.
Thank you.
Okay. Yes. So let's run this. We haven't run this last code yet. Basically, it groups a cell by the column origin and identity. And you can see that we have three colors, or three groups of colors emerging. One for E14, one for E16, and one for E18. And you can now appreciate how you can quickly switch between identities without changing the default identity value. So it's very convenient. We will try this a little bit more later on. There is this next part that I want you to try on your own. Make it two parts actually. Try on your own. I'll paste the code and the link in Etherpad, but optional parts. Right? Basically, I want if you want to give it a try, perform the regressing of cell cycle genes, as well as determine dataset dimensionality. All right. For the first part, code will be there, so you can run it when you have a bit of time. The second part, I don't have the code, but there's a link. It's very simple, a set of code, I think 4 or 5 lines. But what it does is that for the first part is that it determines the cell cycle state of your cells and try to remove the genes which contribute to this the state of the cells. Okay, imagine you have, let's say, just put it simply, you have 2 cells exactly from the same population, exact same region, but they differ based on their mitosis stage, right? You will expect that the 2 cells will be very close together on a PCA plot or UMAP plot, but because the state of the cell is slightly different, some of these cell cycle genes are expressed more in one of the cell than the other, you will see a separation of the cell into 2 groups, which is not desirable. If this is part of your study and you're trying to separate cells by cell cycle, that's fine. You can consider this, and this will be later on clustered as 2 separate groups. But if you want to remove this dependency on cell cycle genes, you have to perform this first part, which is to remove all of the dependency of the cell cycle genes so that the position of the 2 cells relative to the the PCA plot and UMAP plot is closer to each other. So they will be grouped as one cell, independent of their cell cycle stage. I hope that makes sense to you. I don't want to show the, I mean, it's fine. You can run this right now, just that because you have to recalibrate the matrix and renormalize, it will take a little bit of time. And I prefer to show you the downstream part, which is a little bit more important in this workflow. It's more like a side, just as a tangent to the workflow, but it's actually very important, and I use that almost every time to remove the cell cycle dependency of the cell cycle genes. The other one, part 2, determines the dimensionality of the dataset, meaning so initially we have like 28,000 genes, and we reduce it to visually 2 components, right? PC1, PC2. But essentially, you do have a lot of the dimensions right underneath it that is not used for plotting, but you can actually plot all the other dimensions. So what this thing does is to figure out exactly how many dimensions are being considered as principal components, and you can actually use all or just some of the dimensionality for downstream purposes. I'll show you this when we run the UMAP in the next section. But if you use more dimensionalities, it means that you are taking into consideration more variable genes, and therefore your clustering, for the dimensional reduction will be a bit more accurate. So if you understand your dimensionality, you use the right value, you can actually get better separation, get better segregation. Maybe we can try it a little bit later on when we run some of our code and see how it differs. Okay. So just these 2 parts, try it on your own. Okay. So we are done with PCA plot. you've seen how does the PCA plot looks like. We are going to perform the nonlinear dimensional reduction. So this is part 7 now. So this is non-linear dimensional reduction. I'll say both UMAP and t-SNE. But you can choose actually, unlike PCA, which is mandatory, you can choose to run either one and plot either one of these dimensions on your plot. So the function is run UMAP, and similarly, as what you've seen before, we're going to override our previous objects, Seurat object, and you can see how this is useful because you don't want to create 10 objects for each function that you run. You want to keep just one object as it is, and later on, save one object, in for reference, or you can load up this object again later on. So the function is run UMAP. I'm going to start with UMAP first, and the object is Seurat.merge. And I'm going to use, as I mentioned, the dimension. I'm going to use the maximum dimension that you have in this data set, which is previously when I tested this, 30. So I'm going to use all from 1 to 30. You write it as it is. So use all 30 dimensions and turn on verbose. So verbose, as I mentioned previously, prints out the processes within this this function, so you can see how the run UMAP actually works and whether is it completed. So run this in console, and let's wait for a little while. Again, it shouldn't be too long. Data's not too big, and it's just very, very optimized for single-cell RNA-seq dataset, which contains a lot of cells and a lot of genes. So yep, and it's done. Barely a minute. So our UMAP is done. Optionally, you can run t-SNE as well. I'm not going to run it, but you can run the t-SNE. So this is be careful about the naming, because the naming. Let me see, where is the capital t-SNE Seurat.merge dim is equals to 1 to 30. Okay, you can try this out if you want to. Again, it will not overwrite. It will be kept in a different what do you call it, in a different slot in the Seurat object. So it's separate, and you can call out UMAP or t-SNE as and when you want, and we can visualize it. So because I only run UMAP, the default setting or the default dimensional reduction is UMAP. So if you now plot dim plot, the same, capital P, dim plot, the same function that we typed earlier on when we want to run our PCA without any other arguments, it will now be converted into a UMAP plot. All right. So as you can see, the plot looks a little bit different. It's no longer condensed into 2 sections. It's slightly more spaced apart and more clustered together, which is nice. If you have performed t-SNE, there's a quick way to switch between UMAP and t-SNE by providing the reduction argument. So reduction equals to t-SNE as such, right? So if you have run t-SNE, you can do this. I'm not gonna do it right now. And similarly to the previous dim plot, you can also group the cells based on the identity. So I'm not going to comment this out. I'm going to run dim plot Seurat, too much bracket. Again, all the codes are in the Etherpad if you want to follow along. Group by ORIC, equals ORIC ident as such. Okay, so instead of labeling as cerebellum, it will be labeled. It will label the cells based on the experiment E14, E16, E18. Okay. One thing that you can, or you will notice sometimes at this stage, even at this stage, is that if you have dependency on cell cycle stage, you might see that cells from the different stage do separate a little bit from each other. In this case, it's not so evident because you can see this cluster of cell which consists of cells from the 3 different stages. So to me, this is still well segregated based on the salinity, but not based on the stage or the cell cycle stage. So this looks okay for now. But if you start to see that your different samples are pulled away from each other, make sure you run the cell cycle regression before you continue. Okay, so now let's start to plot out, because I think this is quite nice. And usually, if you are able to master the plotting, you can actually create very high quality plots for publication with very minimal modification. So I'm going to show you a little bit of modification that you can use on not only dim plot but other plots as well, and get this data or this plot looks a little bit prettier. So I've shown you the groupings by origin, and I'm going to reuse this grouping for the next few part. I'm just going to copy paste this and add on. I'm going to copy the previous code and add on the arguments to that. So what can you do? You can actually label the cells. So we can label based on the identity. So cells which are around E14, right? Put a label there to represent E14, E16, put a label there. It might not look so nice here because all our cells from E14 are spaced apart. But imagine if you have clusters, this will look nice because each cluster will be labeled separately. So function is label equals to true. And I will also add repel equals to true, because repel basically ensures that the 2 labels do not overlap and there's enough space between each other. So this is roughly how it looks like on this plot. This is where centrally E14 is, E16 is centrally here, E18 is centrally here. Again, I say centrally because this is like a central based on all the cells that you have. And they are so sparse, this is the best position to label these cells. You can remove the redundant legends here. So since you already plotted the labels, you can choose to remove legends. Legends you can add on. I'm gonna pull out this a bit so you can see my screen. Function is no legend, like such. So you add on additional functions to the dim plot with the plus sign. Right? Remember, it's a plus sign, not a pipe or not a other sort of like operators. So a plus sign, run this, you can actually control enter, remove the legends very easily on your plot. You can change the size of your dots and the size of your labels relatively easily. Again, I'm going to copy paste the previous code. Point can be changed using the argument point pt.size and change it to, let's say, 1 to make it a little bit bigger. Run this. So it looks a bit ugly right now, but you can see that you can modify this depending on the dimensional of your figure that you're trying to create. Change it accordingly. You can also change the label size. So instead of pt.size to label.size, let's set this to 10. So now the label will be slightly bigger. Oh, sorry, 10. Yep, as such. So what else can we change? We can also change the color. The cool thing about dim plot is that there is an argument within the function to specify the colors. But this depends on the number of groupings that you have. In our case, it will be easy because we only have 3 groupings. So we provide 3 colors. If you have, let's say, 20 clusters and 20 groupings, it will be a little bit more tedious to provide individual colors. Rather, I would recommend you to provide a color palette or a spectrum of preset colors that comes with R. I think I'm going to show you in the next code. But if you have small number of groupings, you can do calls, short for colors, equals to a list or a vector of 3 color names. Let's do black, gray, and red. I'm going to press this enter so that it prints on the next line so you can see what I typed. So it's essentially calls equals to C bracket, which is a vector that consists of 3 colors, black, gray, and red. So it will print out as it is. The order will be depending on the levels of your identity. So in this case, we start with E14, E16, and E18. So the order is as such. You can change the order if you know how to change the levels of your column. So you can put E16 first to modify the factor levels. So if you know some preset colors, for example, I think previously I've shown you this color palette from our color brewer. They do have default palettes, and you have to just specify the name of the palette. You can do so. So I'm going to copy paste the previous code, and instead of providing the 3 colors, so remove the entire thing. So just calls equals to something, and type in with open with the commas. Let's try set2. With open with the commas. So this is a color palette from there, and you can see the color palette. The first 3 values, 3 colors on the palette prints out as the color of your groupings. Cool. Just more spaced out. The next one is a bit advanced, so I'm not going to explain the code. I'm going to tell you what it does. It's quite cool actually, but the code itself inside is a little bit complex because I use data frame subsetting to basically highlight certain cells. So what I want to do is to basically label or highlight cells from E14 and well, not necessarily dim, but just change the setting, change the highlight color or change the size of the point specifically for that group. So if you imagine if you have, let's say, a wild type and a control, set of cells, you want to show one but not the other, or you want to highlight one not the other. There is a way to do that using dim plot. The code has been pasted, so you can try this out together with me and run this to basically appreciate the output. So this code here on the second line that says cells.highlight allows you to input a list of cells that you wish to highlight. The thing is, you have to know how to fish out, how to extract the names of these cells. And the way I did it was to reference the metadata column. So if you sit down and read this part of the code, you will figure out what I'm doing is basically to use filtering to filter cells from E14, and that's exactly what is being shown here. Cells from E14 are highlighted in cornflower blue, and the size of the points is size 1, and the rest is actually the default of 0.5, I think. All right, so this is quite cool. I'm going to write this as advanced modification. So it might be useful later on if you want to separate different cells in your plot. Okay, cool. I think that's about it about dim plots that I want to show you and the modifications. I'm going to now move on to feature plots. This is similar to dim plot, just that instead of plotting the groupings or the discrete variables that you have in your experiment, you're going to plot out exactly the expression of genes within your set of cells. All right, and you can specify which genes you want as such. So function is feature plot, so it's slightly different. It's feature plot because we're plotting out features. Specify again the Seurat object, Seurat.merge, and this is followed by one or more values or a character vector with one or more values. So you can, if you specify just one gene as such, let's say I want to look at expression of raw alpha, which is a marker for Purkinje cells. You can just type it as it is, and it will basically print out the expression of these cells relative to the other expression of this gene in other cells. So those dark purple color cells represent cells with high expression of raw alpha, and those gray color cells do not express raw alpha at a decent level. If you have a list of genes that you want to plot out at the same time, you can do that simultaneously. So instead of providing just one gene, provide as many genes as you want as a character vector. So C, specifying the character vector, followed by a list of genes. I'm going to try raw alpha, I'm going to try PAX2, I'm going to try PAX3, A to1, this is granule cell marker, PAX6, and LHX9. Again, code is already in the Etherpad if you just want to copy and paste. Right? Cool thing about this is that it uses this package called patchwork, and it will patch all of these individual plots together into one plot. So in one view, you're able to look at the expression of all of these marker genes in one go, and you can identify some of the cells.
Yep. Can you also add the information or group by the original identity?
Yes, you can. You can. You can do a little bit more modifications. There are a lot of options here. I think one of them is depreciated, but let me see. So if you do this, I basically, type comma followed by tab. There are a couple of arguments here. I think split by is one of the arguments that you want. It does give you the ability to split by some sort of identity. I think there's a group by as well. Let me see. Or maybe that's for violin plot. There is another. Yeah. You can do split by. So let's say let's try this. I don't make it too big, but you can basically split by if you provide oric.ident. Let's try this out. I don't typically do this because we're looking at the like the expression of these genes as a whole. But if something that you want to look at, this is exactly what you want. It looks a bit weird because it separates the three different or the identities, which is exactly what we are trying to do. And then for each gene, you look at. Let's try raw alpha and see how does it look like. Just copy and paste this. And just type raw alpha.
Yeah, I got it. Yeah.
Yeah. And you see that you can split it. I don't know how well it looks. Yeah, so some cells will appear, right? So like E14 and E16, the number of cells, like population of cells is different because you're only putting cells from E14 here, cells from E16 here. So you split them up based on the identities.
Okay, thank you.
I guess this makes sense. Cool. Okay, let me see, where was I? Yes, so the same set of modification that you have seen previously in dimplot can be applied. I guess not all, but you can change the point size. You can remove legend if your plot has some sort of scale. You can do that. In this case, we don't have, but the previous plot should have a scale. So you can remove the scale if you don't want to see the scale. You can change the color of the expression, providing a different palette or a different range of colors if you wish to. All right. Cool. Last one, last very cool plot, which I like, violin plots. Previously, we saw violin plots when we plot out the discrete variable, not discrete, the content variable, the number of genes, number of RNA counts, and percentage MT. We're going to use the same function, violin plot. but we specify different features right now. So let's do the same raw alpha plot. So you specify again, first one, specify the name of the object, Seurat.merge, and then followed by the name of the gene that you want to visualize. So we do valid plot, and you see this the expression of raw alpha across your entire sample or the entire experiment. Similarly, you can group them. So you can, I think there is a group by for this. Yes, group by do appear here as an argument, so we can change this by oric.ident if you want, so that it now I shouldn't change the previous code. Let me just correct this a little bit. So if you can do oric.ident like as such, if you want. So you can see how this volume plot will benefit you if you are trying to compare expression from different cell identities. So if you have different clusters and you want to see expression of certain genes within each cluster, you can do this very quickly using volume plot. It doesn't look nice here, but let's try to continue on and replot this as we go along. All right, so 3 plots that I've shown: dimensional plot, plotting the PCA or UMAP dimensions of your experiment, feature plot using the same dimension but plotting gene expression, and volume plot gene expression grouped based on a particular identity that you have set. Okay, let's move on. I think this is also the next nice part, which is cell clustering. So if you see from our previous dimensional plot, let me just bring it up a little bit. Let me see, let me plot out a, it should be fine. So, yeah, so if you look at this, you can appreciate that there are certain clusters appearing. It might not look, as condensed or as well segregated right now. If you do the cell cycle regression, it will look a little bit better. But for now, you can see certain groupings. For example, this part here looks as a cluster. It appears to be like a cluster. This appears to be like a cluster. Even this small set of cells within each cluster do represent individual clusters. And you can actually run functions to identify all of these, not identify, label or separate these clusters from each other and group them according to its position within the dimension. So before that, we have to run this function called find neighbors. So essentially, you figure out which cells are neighbors to each other, and then based on this information, group them into clusters. So 2 functions. We're going to start with find neighbors first. Seurat.merge. And run find neighbors, followed by the object, followed by the dimensions that we use the same way as we specified previously, dims equals to 1 to 30. All right, so we're going to use the, not maximum, but as much dimensions as we have to identify these neighbors. So give this a go. It will take some time. And then we can proceed on with the next function, which is find clusters. So same thing, override the name of the previous variable, followed by the name of the function find clusters, and the object that we're going to use to find the clusters in. And I'm going to add another argument here, which is called resolution. And resolution here indicates, basically it's a numeric value. It can take up a float or a decimal point value, and it tells you, okay, in layman's term, how many clusters do you want? Do you want to have a lot of clusters and separate them finely, or do you want to give a coarse clustering and just give lesser clusters, but very coarse number of cells in each cluster. So the lower the value, the bigger the size of the cluster. So you have less cluster, but bigger cells, and not more cells in each cluster. A bigger value of resolution gives you more clusters, right? We're gonna start with 0.2 because I don't want too many clusters. I wanna show you the clusters based on a very generic markers, which I've determined previously. So I'm going to start with 0.2. You can go up to 1, 1.5, or even 2 if you want to have more clusters. So you can play around with this. If you run this, it will do two things. It will label your cells based on the cluster that it identified, and you can actually see this from your metadata. If you were to print out your metadata now, Seurat.merge at metadata, and just basically head this so that it looks a bit more prettier. You can see a couple other columns. Actually, we have moved quite a lot actually. You can see that now there are two columns, SCT underscore SNN underscore rest at 0.2. This is the output of the clustering that we have performed. So it will label each cell based on the clusters that you identified. And then it will also replicate this into another column called Seurat clusters. The reason why they did this is because they still want to retain the clustering identity depending on the parameter that you use. So if you change this to a resolution of 1, you will create another cluster with a different value based on the resolution. But Seurat clusters will still be there because this is sort of like the assigned or default clusters that you have assigned. So it will create these two columns and also refresh or reset the identity of your cells to Seurat.clusters. Let's try doing a dim plot. All right, just a general dim plot on Seurat.merge, and you can actually see what I mean. Run this, and instead of previously we just see cerebellum as the grouping, now it has grouped the cells into 12 clusters as such. Right? Right, so create clusters and then assign the default identity to this newly created cluster called Seurat clusters. But this is not fixed. You can still change the identity again as how we have done it previously. You can always manipulate the identity and modify it according to your desired analysis. Okay. Let me see. So if you were to do this, repeat this and label the clusters so you can actually see exactly where each clusters are positioned. You can do label equals to true, and then maybe we can repel it as well. Repel equals to true, and then do no legend. It's a bit redundant here. Just to appreciate that you can actually see labelings for each cluster, and this is very pretty just to begin. Even though your data looks less optimal, when you see colors of a spectrum and segregated from each other as a sort of UMAP plot, it gives this sort of euphoria, that you're doing something correct, and drive you to kind of like finish out the whole analysis. That's at least for me. This looks very, very nice, very pretty. But then this is not the end, because you can proceed and figure out the identity of these clusters. So can we somehow use or identify the markers for each of these clusters to figure out the identity of these cells, right? So this is exactly what you want to do, and then use that list of cell markers and compare it with a list of cell markers that you know or previously published, so that you can assign the identity on your own, or you even use some sort of machine learning algorithm to identify the cells for you. I will show you an example later on a link to perform this. But essentially, this is very powerful because it allows you to do a lot of downstream experiment, a lot of downstream analysis using the information from each gene markers. Okay, I'm gonna just relabel this as a new part. I lost count, probably part 10, identifying gene markers. Okay, so there are two ways to actually find markers. You can do this globally and use just one function to find all markers. The function is called find all markers as such. It's called find all markers. And then input is Seurat.merge as such. You don't run it first because it's going to print out in console. And you don't want to do that right now. This will find all markers for every single classes or every single groupings that you have. So if in this case you have 13 clusters, not 12, 0 and all the way to 12. So 13 clusters, we will find markers for each of these clusters and output a data frame that tells you the genes for each cluster and the sort of matrix, the log4 change, how many percentage of cells have this marker. So you can actually filter them out a little bit more later on. Or you can actually run another one, which is find markers. Sounds similar, but find markers allow you to refine the comparison and instead of running the whole the whole loop, you can specify which cluster you want to analyze and comparing to which other population. We're going to do both, but we're going to use the first one to find markers, the second one to identify differential expression in the last part. All right, let's run this. Let's do this first. As I mentioned, the output is a data frame. The thing here is that it will not be saved as a slot in your Seurat object. It's actually a data frame on its own. So instead of, this is the only part that you have to create a new object and not reuse the previous object. So if you reuse the previous object, it will overwrite that variable name and you have to start all over again. So take note that you have to create a new variable. So cluster markers will be the variable that I'm going to create. So you will save the data frame into cluster.markers. And I'm going to provide a couple more arguments. We can follow along. First one is only.pos. So select markers which is positive in that particular cluster. So set it as true, only.pos equals to true. You can put T if you short for true, or spell out the entire true if you want. Yeah, it doesn't really matter here. The other one is min.pct. This gives this metric basically select cells which have a minimum percentage of cells in that cluster with that gene of 25%. So if you have, let's say, 1,000 Purkinje cells, right, that particular marker, for example, raw alpha, has to be at least 25%, in at least 25% of those cells. So 250 cells has to have this. You can play around with this if you want to create a more stringent or more, more relaxed parameter. I think default is 0.1, but 0.25, or making it slightly higher, especially for finding marker genes, is slightly better. Last one will be log fc.threshold. So this is the fold change threshold between the two clusters. So if you look at cluster 1 versus all the other clusters, so this is how you compare markers, right? 1 versus others, the difference should be at least 0.25. So this is at the linear scale. So at least 25% increase or 25% more in terms of expression as compared to others. So there's two thresholds, the percentage of cells expressing this, as well as the fold change of the expression between the two groups. Okay, I'm going to set this as 0.25 as well. I didn't mention it earlier, so set it 0.25, and let's run this. It will take quite a while if you have a lot of clusters. We have 13 here. It shouldn't take longer than 3 or 4 minutes, it should be okay. But just give it some time, and then we can basically talk a little bit more about the other part. If you are dealing with or you're trying to identify gene markers, do look through the data frame clustered up markers and plot out the values on your own. Basically, ask yourself whether or not the threshold or the filters that you use is good enough to separate your markers. For example, if you know that you have a particular marker, for example, again, raw alpha for protein itself, if you know that this particular marker should appear, and you want to make sure that you're selecting those highly confident markers, bring the threshold, let's say mean PCT or fold change, a bit more closer to some sort of known markers that you want to look at. There's no right or wrong here. I had this conversation with a couple of people. There is no one answer. I cannot tell you to choose, let's say 50% or one fold change. It's difficult to set a particular value because different experiments require different filtering parameters. So you have to eyeball this, look at your data set, plot out, basically use certain reference genes and see if the values are more biased towards that cluster that you expect it to be. Right? It does take a bit of practice. I would say that I'm not the best at this as well. I do make mistakes sometimes of using the wrong parameters and have to do it again and again and again. But that's the whole point. You iterate the whole thing to refine your list, and hopefully the list has a decent size, a decent confident gene that you can actually use for validation or downstream analysis. Right. I think we are almost like 3/4 way. We have a couple more. Let me see. I can talk a little bit about differential expression first. So part 11, while this whole thing runs, differential expression. So this is very similar to the find all markers function. You provide the Seurat object, Seurat.merged. You will not run this right now because there's a background code running, so don't worry about accidentally pressing anything. You will not run, but you can modify the code because we are working with a script. So that's nice that you can wait for your code to finish and continue on with your script. So the two arguments that is important here are the identities of your two groups. So you are comparing two groups of cells. So you have to provide the identity of the first, followed by the second. And the two arguments that you can actually use to do this are ident1 and ident2. Ident1 will be the sort of the group that you are focusing on, and ident2 will be compared against ident1. So for example, if you're trying to find markers, like as from the previous code, find markers for the first cluster. So ident1 will be cell from cluster 1. Ident2 will be cell from all other clusters. So that's the idea behind finding all markers. If you just want to compare two groups, you can provide specifically one for the ident1 and another group for ident2. I initially specify ident1 as 4 and ident2 as 7, and this is based on a previous, not 7, ident.2 equal to 7. This is based on a previous workflow that I use. Hopefully it still looks okay right now. So this will compare to the two groups, cluster 4 and cluster 7. Imagine that you have different sort of classification. Let's say you have cells coming from a wild type mice and comparing it with a treated mice. You can specify different identities for the different comparisons if the identities are present in your metadata. That's very important. So in our case, we have the original identity E14, E16. We can use that. If you want to add on more metadata, you can do, we can create more columns to separate the cells a little bit more finely and specify the entities as such. So the values here can be anything as long as the values are in your metadata. And also it's the default identity. So in this case, our default identities are these clusters. So you can actually use the values 4 and 7. Okay, so the cluster markers is done. I'm going to I'm going to modify this code a little bit so that we can actually see top 3. We're going to look at the top 3 markers of each cluster. All right, I'm going to modify the code and I'm going to paste it into Etherpad, and you can run it. I'm just going to run this as well so that we can quickly just visualize the top 3 genes for each cluster. So I already pasted it. It's called it will create a new variable called top 3, and it has these 3 lines of code, a combination of piping and finding out the top 3 genes from each of these cluster, and do run it, and we can have a look at the data set. I'm going to view the entire table of this top 3 variable in a new viewer. So I'm going to type view followed by top 3. Well, I shouldn't type it here. Let's do it here. View top 3 as such. Remove this, a bit weird, right? And do run this in console so that you can actually open up the entire table as a viewer, and you can actually see cells. I mean, not cells. We can actually see the markers, the top 3 markers for each cluster. Okay, just a bit of explanation here. As we have mentioned previously, you have this second column, which is the log4 change, the average log4 change between that particular cluster, cluster 0, versus all other clusters. So this is what it means. This is the percentage of cells expressing this gene. So this gene FAP-P7 is expressed almost 100% in this cluster, but only 26% in other clusters. So this met the previous threshold that we set of a minimum of 25%, and it's very clear upregulation as compared to other clusters, which is good. 1, 2, 3, 4, 5. The 5th column is the p-value, the adjusted p-value of the comparison. This is adjusted from the first column. It's all 0 here because it's highly confident statistics. So this is very clear marker genes for these clusters. And then you can see the next 2 columns specifying the cluster ID and the gene that it identified. Okay. If you're familiar with your population of cells that you're working with, and you can identify these marker genes by eye, that's great. I can see a couple. I think PCP4 and CAR8. These are, not mistaken, these are Purkinje cells, if I'm not mistaken. I think so. Yeah, I think it should be Purkinje, different stages of Purkinje cells. Let me see. VTN is very familiar. VTN is, I think, meninges cells. Claudin 5 is endothelial cells. There's photostatin here as well. There is, I was looking for raw alpha, but probably it's not the top 3. So we chose the top 3 genes. If you change from 3 to 5 or 10, you can see more. Marker genes for each cluster. Probably it's a bit too little. What else can we see? Neuro D1 is granule cells. So there are very clear what do you call it, markers for each of these clusters, which is cool, which is really enriching, so that that you are doing something using the right pipeline. One thing, this is weird. I've seen this a lot of time. This gene called exist. It's one of the 2 long non-coding RNA that is differentially expressed in males and females, and I always see this coming up in different experiments. Probably there's a bias between the number of male derived cells versus the female derived cells. So so that's why it sort of popped up and very, very confident, very highly expressed in cluster 4, but not in other clusters, which is weird. And then yeah. So you can look at this. If you want to eyeball your list of genes, you can change the parameters again. You can run again to find all markers if you want to make the identification more tighter. You can also, another thing, filter this list further by actually comparing the two PCT1 and PCT2. So currently, the difference is quite large for the first gene, but the fourth gene, it differs by about 25%. So if you want to find something which is, like highly expressed in one, but very lowly expressed, like for example, the third gene, this is very clear difference. So you can use dplyr or other manipulation, data type manipulation to select select genes which is highly expressed in one, but very low expressed in the other one. That's another tip from me. Okay. Couple more things. We're about done. If you want to view a heat map of the expression of all of these markers in one view, you can do so using the function do heat map. Similarly, the function requires the Seurat object followed by the features. Instead of using all features, we're going to specify the features or the genes from the top 3 marker genes. So the function is top $3 top $3 genes as such. So we're looking at just couple of genes, so it will not take too long to print it out, and it will not be too complex for us, and maybe just give no legend so that we do not see the scale. It's plus no legend. Oops, and try it out. I'll run this in console. The thing about heat map is that it might take a while depending on the list of genes, and do make sure that you know the number of genes that you have in your list, and not print out like hundreds of genes, because then it will take too long, and also just stop your computer from doing anything else, because it's channeling all the resources into printing out that heat map, and I don't think I will want you to undergo that.
A question?
All right. Yes.
A question. If I were to just have the heat map of, let's say, cluster 4 and 7.
Yep.
How can I add that information here?
Yep. Same thing. Arguments. Let's look at arguments in a while. Okay.
Arguments, identity, one.
Yes.
And
let me see.
okay.
Let me see. Again, I don't usually do this so often, but let's see. We have let's look at the arguments. So let's see. You can provide the cells to plot. In this case, it's gonna plot all the cells. If you know which cells you want to plot, if you remember the previous advanced modification that I showed you earlier on, if you follow that same code, you can actually select cells that you want to plot. You can group all these cells based on a different identification. So currently it's based on clusters. You can change this to the original identity, E14, E16, if you want to. It will not look as nice, because the genes that we plot out are markers for each cluster. So that's why it looks. So basically what I want to say here is that this looks very representative of marker genes, because let's say the first 2 genes are highly expressed in cluster 0, but not in other clusters. And this is like diagonal line that labels highly expressing genes like in this way. So you can see the marker genes as you go from left to right, corresponds to each cluster. So if you were to plot, or if you were to group using a different identity, for example, if you group by the auric ident, let's say like that, it will look different, because your markers might not be representative of that identity, but it will look a little bit more sparse, right? What else?
I can group by identity. Also the clusters?
Yes, it's already clusters right now. It's already from 0 to 12, right? So this.
Let's say I want to visualize just cluster 2 and 3.
Yep. Let's see. Let me see. Group by. There's another way of doing this. Let me see if there is a. Yeah, probably they don't have this here. Angle. It's just display mean group colors. Okay. I was expecting some sort of like ident, like to provide a vector of ident. But if they don't have, it's fine. These are all still R objects, and you can do a lot of manipulations to it. One thing that I can recommend is this. So let's say this is very specific to a request to plot certain clusters. So one thing that you can do is to subset the data frame, or sorry, subset the Seurat object on the go. So previously we use subset to subset cells which are passing the quality control, but you can also subset based on other metadata. For example, you want to subset equals, let me see if I can get this correctly. This is Seurat clusters in C4 and 7. Right. So what it's trying to do, again, you don't have to understand, you don't have to kind of like comprehend this code, but specifically it's going to subset this Seurat object first before plotting the heat map. And it's going to subset based on the column called Seurat cluster. I think it's cluster or is it clusters? It's clusters, I think. Column Seurat clusters, and select values which corresponds to 4 or 7. All right. If you run this, it should be a bit more faster, and you should only see the two clusters. Let's see, like that.
Oh, perfect.
All right.
Cool.
So it's not pretty because we are actually modifying the original Seurat object on the go, but it does give you what you want, right? There is another way. Let me see. What I thought of earlier on. Yeah, can slip my mind after I spoke about this method. But yeah,
okay.
Let's go with this first. Probably there's a update to Seurat functions, and you can actually do this a bit more better. Or if anybody else knows a better method, you can voice out right now. I know there are other packages that can actually plot heat map a bit better. One is called Silus. You can try this out. The package is called Silus, but you have to find the package, the name of the exact package that is used to download into our Studio. So you can try this out. It does a better job because you can group not only by one grouping, not only by clusters, but by clusters additionally, let's say another variable. So you can basically have a more refined heat map that you want to use for publication. So it's quite cool.
Okay, thank you very much.
Yep. Let me see. Yep. Okay. So I know it's already 1 p.m. Just one last part, just to finish up the differential expression. So I'm not going to run this, but you can basically run this code on your own. Again, assign it into a new variable. So this is let's say cluster 4 versus cluster 7, right? And find all markers. And there are again a lot of other arguments that you can actually provide. For example, this min.pct is still there. This is the min diff.pct, which allows you to select based on the difference in the percentage of cells. The for change will also be there. Not for change should be somewhere there as well. Yep, not for change. So you can add on to the function to refine your list of genes as well, and you can plot out. You can view all of these list of genes using heat map as well, or using volume plot or dimensional plot, as how you are you're very familiar when you look at papers that plots out single cell RNAseq data. Right. Yeah. I think that's about it. That should be the last part, because volume plots, I'm not going to show the volume plots because I've already seen how we do volume plots previously and all the arguments that you can actually give to the function. So I think we can stop here. There are a couple of things that I want to basically add on, some information for you to sort of bring back. The first is you can actually use the same workflow for single cell ATAC-seq. It does use the same, some sort of same pipeline and same syntax, and it works okay. But there are other tools that you can actually use. I want to mention just two. One is ChromeVar. I pasted it in Etherpad, and the other one is Archer. These two are currently one of the better tools for single-cell ATAC-seq data. So try to use that. It looks a bit different, but essentially the idea is the same. You go through the same QC, the same normalization and stuff, so you can get a more refined data set for single-cell ATAC-seq. There are other tools that you can also use. Monocle 3, I'm pretty sure some of you are familiar with this. It does nice trajectory analysis. You also have other non-R tools like ScanPy, which runs the same analysis on Python. Does use similar model, but maybe you get slightly different clustering and different marker genes if you use different tools. But essentially, you get the same idea and the same enrichment. Last one that I use recently is called URD. I like this because it identifies gene program. So instead of looking at just one gene and see how it changes, it groups genes together as a gene program and tell you exactly the list of genes in each gene program. It's quite interesting actually. Other analysis that you can actually do, like I said, trajectory analysis using Monocle. If you're into that, depending on the the request, I can try to do trajectory analysis in the future if you want that. If not, the vignette is actually very easy to follow. You can do probabilistic assigning of cell identity. So instead of eyeballing the markers, you can look at all of these marker genes, put it into a machine learning model and get them to assign the cells for you. One that I used quite regularly is called CellAssign. It's pretty good actually. You have to provide your own matrix with information on cell markers as input as well. So that will take some time to figure out your markers. And then you also have, this is another one, the identifying gene program, CNMF. This is non-negative matrix vectorization, another way of identifying gene programs. So yeah, I think we are done. I do have a couple of feedback that if you wish to provide, they are in the Etherpad, and if you want to share with me what you learned today and what we can improve on in this workshop, I'll be very, very helpful. You will really sort of like understand how we did and what we can improve on. So that would be great. If not, we'll call this a day. We're going to take a break from workshops for about 2 months. Probably we'll resume in August for workshops. But if there's a request to sort of like have a meeting so that we can answer specific questions on Seurat or other sort of analysis, I'm open to doing so. If you are available, we can actually set up like a quick meeting just to trash things out. But if not, I wish you have a good day and a decent rest of the week. I'll see you around. Thank you.